So we have the metabolic adaptation during fasting, which is called the glycogenolysis and glycogenesis. So these are all mechanisms. The body will always undergo some mechanisms, some homeostasis in order to maintain the body. So we have uh, short-term fasting and we have prolonged fasting. So during fasting, our body most time does two things, which is glycogenolysis and gluconeogenesis. Glycogenolysis from the word glycogen, lysis. Example, virus kills cells by what? Lysis. So what is lysis? We are breaking down. So glycogenolysis means breaking down of what? Glycogen. So the word glycogenolysis means we are breaking down what? Glycogen. Glycogen, lysis, breaking down. So glycogenolysis is breaking down. Why gluco means glucose, neo means neogenesis means building, anabolizing. Building is anabolizing, while breaking down is what? Catabolism or catabolizing. So in short-term fasting or in fasting, whether short-term or prolonged fasting, we usually undergo glycogenolysis and gluconeogenesis. That is why when you are fasting, you can never die. So do not be deceived. When you are fasting due to spiritual purpose or some other reason in your life, you will not die until all the glucose molecule, carbohydrates, proteins, and every other molecule in your body has been drained entirely. That's when you will die. Starvation. So during starvation, protein starts serving as what? As source of energy. So fast, it usually helps the body. So we have liver glycogen. So the glycogen is the liver and it's to undergo what? Glycogenolysis. Remember, glycogenolysis is what? Breaking down of what? Glycogen. And when we break down glycogen, we form what? Glucose 1-phosphate. And when this glucose 1-phosphate, we now convert this glucose 1-phosphate to glucose 6-phosphate. As we convert glucose 6-phosphate as we convert glucose 1-phosphate to glucose 6-phosphate, we now take it into the what? Into the smooth endoplasmic reticulum. At the smooth endoplasmic reticulum, that is where we have glycogen phosphorylase. And what does glycogen phosphorylase do? The glycogen phosphorylase releases what we call the phosphates from the glucose, forming what? Glucose. This glucose now become bidirectional and move back to the blood in order to maintain the blood, in order to help to maintain the blood level, the glucose level in the blood. So that is for the short-term fasting. So in the prolonged fasting, something is happening and it is very slow. The process is very slow because we are breaking down triglyceride or lipid, and it's very slow, which can take up to 12 to 14 hours, but it releases huge amount of energy. It releases energy of about 100 and something ATP, about 100 and something ATP, ranging from 108 to 135, so depending. And in the body we have, in the muscles we have lactates, and glycolysis is the conversion of glucose to pyruvate, and this pyruvate is converted to lactate in the absence of oxygen, and pyruvate is converted to what? To acetyl-CoA in the presence of what? Oxygen. So pyruvate is being converted to lactate in the presence of oxygen. This most time when you're doing vigorous exercise, when you're doing massive exercise, example like playing football or running for a very long time. What is happening? Your body will keep on converting pyruvate to lactate, and during this process, NAD positive During this process, NADH is converted to what? NAD positive. So this NAD is called nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. So this is the reduced form, H reduced form, while the NAD positive is the what? The oxidized form. So we are converting NADH to NAD positive. The process by which we are converting what? Pyruvate to lactate. So we are converting pyruvate to what? To lactate. We are converting pyruvate to what? Lactate. And in this process, ATP is what? Produced. 2 moles of ATP is produced by converting ADP to what? To ATP. 2 molecules of ADP is converted to what? 2 molecules of what? ATP. And this is called the Cori cycle. What is the Cori cycle? The Cori cycle is the process by which lactate is converted to glucose and is not taken back to the muscle. So in this process, the glucose or energy in the muscle is broken down to give us alanine and lactate. This will now further go and convert to what? Glucose. So the enzyme that stimulate this are fructose bisphosphatase and phosphoenolpyruvate carboxylase. You understand this better during when you are going to talk about glycolysis. So maintain stable blood glucose through hepatic glucose production. But let me explain something to you. Fructose bisphosphatase does a very specific function and important function, and phosphoenolpyruvate carboxylate also do the same thing. They do a very good function during gluconeogenesis, which is prolonged fasting state, and this now leads to accumulation of acetyl-CoA and leads to high yield of energy of 100 and something.